Hello, how are you?
Yeah, fine.
Great. That's good.
I haven't done much.
So, did you have any papers you wanted me to go over? Or the last time that
No,
was pretty good.
I think I might have got one, but it wasn't. I think you mostly went over them last week. The ones like I have a whole binder here, and I have to make notes and then write about them. And make sure that I've got all of the relevant ones,
Yeah.
so that I can be thorough. And then I can add it with what I wrote already, and I might have a paper.
Yeah.
So I'm almost there in a way.
Yeah.
I have my references, I have my new references, and then I have written half of it, which is just a summary of my thesis, but
Right.
yeah, a very brief one. I'm not explaining how tensegrity works or anything in it, just sort of an overview of how it works. But it would be nice if there was some software out there that would do tensegrity, because, like, the entire human body, from macroscopic to microscopic, is our tensegrities. They're all in tension. So
Right.
some sides get compressed, but basically they're hanging, like hanging on to each other,
Yeah.
attached to each other. So yeah. How come you had so many last week and this week there doesn't seem to be many?
I mean, people or
Yeah.
I don't know. Well, I think uh, Leilith wanted to present on his work, and he had his friend here. So, I mean, I don't know.
Yeah, that was nice. Yeah.
Yeah, I don't know. Well, anyways, I have some things planned here I wanted to go over. Actually, a nice paper on something called the Cellular Olympics. And then, yeah, we'll see what that is. And then another paper on these structures that are like self-organized by insects. We'll see what that looks like. So,
Okay.
yeah, I have these two papers. Let's go over it. Yeah, this is the Cellular Olympics. This is from Annual Reviews of Microbiology. And the subtitle is Ultrafast Cellular Motility Across the Tree of Life. So this is Ray Chang and Manu Prakash. Manu Prakash has done a lot of interesting things with difference in cellular biology in general.
Oh, I
So
wonder if you've done any tensegrity.
I haven't
Oh,
seen. Yeah, might look over his publications, but I doubt it. Um, this is on like this is biophysics and actually cellular ultrafast biophysics.
oh,
Um, and in ciliates and dinoflagellates.
it's funny. The development of the axolotls is ultra slow biophysics.
Oh, really? Yeah,
Well, yeah, it happens over hours, like 2 days
yeah, yeah, yeah. You
or
have to
longer.
really kind of devote a lot to imaging for that, like just getting maybe like a real time account of it.
Okay, I maybe while I'm thinking of it, I'm going to do this project where I'm trying to get a baseline on salamander eggs, blastocyst development using optical coherence tomography.
Hmm.
Um, I guess I should ask Dick this too. Like, how often should I image them? If I imaged only 20 eggs, would that be enough? I guess that's sort of a statistical question in a way.
Yeah,
Maybe I'll ask Dick, but yeah, I need to figure that out.
yeah. Okay. So yeah. Let's go over the abstract. So surprisingly, many single cell organisms and specialized cell types can achieve speed and acceleration significantly faster than those of multicellular animals. They're dealing with single cell organisms with ciliates and dinoflagellates, and this is, of course, different than migration in a multicellular embryo, where you have migration during different stages like gastrulation. Cells are moving around, and the embryo itself is changing its shape. But this is single cells, so they're moving around their environment. They might be moving around collectively, but it's always faster than in the embryo. These remarkable cellular machines must integrate energy storage and amplification and actuation, latches for triggered release, and energy dissipation without failure. So you have these aspects of actuation and latching and other types of things, and they have to develop these very reliable mechanisms. So basically have these macromolecular assemblies inside the cell that build these structures. In this review, we first mapped the atlas of single cells across the tree of life that use ultrafast motility. So this is they're proposing, I guess, that there's some clade where ultrafast motility has evolved and has these kind of mechanisms for ultrafast motility, very high performance, high accuracy mechanisms. We then quantitatively compare extreme acceleration, speed, area strain rate, volume expansion strain rate, and density change rate among single cells. So we have these different measures. We have extreme acceleration, which is just acceleration, but we have to have a technology that can capture the speed of, the movement without aliasing. We have speed, which is, of course, measuring the displacement, as opposed to acceleration, which is a derivative of position, like the second derivative of position instead of the first. Area strain rate, which is, of course, something that Susan is interested in, where I guess you measure it over an area. Volume expansion strain rate, where, again, this is just kind of like the difference between just area and then volume expansion, or sort of expanding that area. And then density change rate, which is, changes in density, I guess. And this is all among single cells. So you need these different types of measurements, and you need to be able to derive them from microscopy. Next, we generalize these ideas by placing various trigger, actuation, and dissipation mechanisms within a unified framework. So we have these different mechanisms: trigger, actuation, and dissipation. So trigger would be like, I guess, something that kicks it off. Actuation is something that helps it move. And dissipation is where you have like a release into the environment. We conclude with a detailed summary of the diverse functions enabled by ultrafast cellular motility, providing a comprehensive foundation for understanding extreme biophysics and its diverse role at the cellular scale. So this is, again, this extreme biophysics. It requires specific metrics. It also requires, a good type of microscopy, the appropriate type of microscopy, and then it requires these kind of mechanisms and thinking about them in a theoretical framework. Okay, so like a lot of these annual review articles, they have like a table of contents. So it looks like, we can go through this. It looks like they classify behavioral phenotypes for ultrafast motility. So they have hydrodynamic swimmers, hydrodynamic jumpers, elastic jumpers, surface-based motility. That's where they you have these swimmers kind of on the surface versus swimmers within the water column. So hydrodynamic being, I guess, optimized for going through the water column fast. Elastic jumpers that could be just on the surface of the water, where they jump from place to place, or they jump in a certain way, and there's an elasticity involved in that. Then there's, of course, surface-based motility, where they move around the surface. There is contraction without translocation. There's cell protrusions of extrasomes and extreme volume and density changes. So all these things are involved in ultrafast motility. And then quantitative comparison for ultrafast cellular motility. We have ultrafast velocity and ultrafast acceleration. So these are two components of position. So we can get this information from positional data over time. If we sample the trajectory dense enough, we can get velocity and acceleration, but we need to be able to get that information at a high enough scale, or a fine enough scale for it to be like accurate. Then they have trigger mechanisms of cellular ultrafast motility, actuation mechanisms, and energy storage of cellular ultrafast motility. So these are trigger and actuation mechanisms. The trigger mechanism is for cellular ultrafast motility, and actuation mechanisms and energy storage are also for cellular ultrafast motility. So this is a lot of biophysics, internal biophysics. So this is calcium-sensitive ATP-independent non-actin filaments, cataclysmic microtubule disassembly. So this is where you have changes in the, changes in the structure of the cytoskeleton with respect to movement, decondensation of secretory proteins, osmotic pressure-driven mechanisms, and purely hydrodynamic-based mechanisms. So, I mean, this one looks really interesting, 5.2. But then 5.3 also looks interesting. It's just basically a lot of sort of adaptations that the cell needs to make to do this kind of ultra-fast movement. Then there's energy dissipation mechanisms of cellular ultra-fast motility, functions, more general functions of cellular ultra-fast motility, and then some discussion about it. So I actually wrote a couple of blog posts about this like years ago, and it's just kind of thinking about like different sort of movements that you see in multicellular organisms, thinking across the clade of vertebrates and invertebrates. But anyways, this kind of talks about how you see movements in across the tree of life. So it kind of talks about like really fast movements or really fast organisms and really slow, or so actually it talks about very fast movements in animals and in plants. So this is, for example, the cheetah is the fastest running land animal with a horizontal speed of up to 25 meters per second and an acceleration of 1.5 gravity. A grasshopper jump can reach an acceleration of 18 gravity. Plants have also evolved various ultrafast mechanisms for nutrient acquisition and reproduction, exhibiting velocities and accelerations comparable to those of animals. And we don't think of plants as being big movers. I mean, they obviously move, like their cells move out and extrude as they're growing their leaves and they're growing their roots. But we don't think of that as being particularly fast. And actually, as it turns out, there are very fast movements in plants, especially with respect to nutrient acquisition and reproduction. So this is specialized mechanisms, again, like what you see with the cheetah and with grasshoppers, in terms of moving, like limb muscles and limb composition. You see this in plants as well. So in plants, you have the bladderwort suction trap, which can achieve a fluid velocity of 1.5 meters per second and an acceleration of 600 g, allowing the plant to trap water, fleas, and worms for its food. The bunchberry flower has a catapult mechanism, enabling it to eject pollen at a velocity of 7.5 meters per second and an acceleration of 2,400 Gs. As its pollination is self-incompatible and requires this ultrafast mechanism to enhance insect pollination and potentially wind pollination. On an even faster scale, the famous mantis shrimp appendage can strike at 31 meters per second and 25,000 G. So when we say like a gravity, if you think about like um, pilots when they fly a plane and they fly it at 1G or 2G, it's this is like that's sort of the measure here.
That's a lot because
Yeah.
people can only withstand about 10G.
Yeah, it's a lot, and it's interesting because you think, and if we were exposed to those kind of inertial forces, it would tear your body apart. But
Yeah.
these are adaptations where you can evolve biological materials or biological structures, functional structures that exert many, many more G's of force than you would have.
Yeah,
Yeah.
I've always been opposed to centrifuging things like eggs and sperm because I think it kills them.
Yeah. Well,
And there was an experiment done. I was asked to review the article, and they actually centrifuged sperm, and they found that the longer you did it, the less success rate there was in fertilizing eggs.
yeah,
So I said, "Oh, somebody actually did a practical experiment that it works. That it explains why some things in a laboratory don't work. Because
yeah,
they just put it through the centrifuge." I just so I praised the article very highly and sent it on its way.
yeah. I think the rule sometimes is like you can centrifuge RNA and proteins all you want, but like cells, if you put cells in a tube, you have to be very careful because they can't withstand those kind of centrifugal forces. They'll break apart, and so
Well, sperm are sort of alive. Like
yeah,
they've got ATP going on and a structure, and yeah, you can wreck that.
yeah. Which I
So
mean,
it was just,
yeah, which begs the question: why did these cells and these types of structures survive these kind of forces?
imagine you could work on some engineering if you could understand the whole mechanism there. I think that would be great. Like you could design your micro machine to propel something if you
Yeah,
could study those mechanisms
yeah,
and the protections around those mechanisms so you won't wreck the wreck the machine.
yeah,
Yeah, this is a very good and interesting thing. It leads to other other things.
right, right. Yeah, it does. So,
Yeah, at least look at the papers and find the flower and study them.
yeah. Well, now there's some other organisms. So, like a lot of times you'll have organisms where they have a specific function where they have like a ballistic movement that is very fast. And you can think of like a frog's tongue that goes out and gets like an insect. It shoots it out and brings it back in. It's very fast, and that's a lot of force that's being generated. And then it isn't just kind of like put out at the onset. There's a control there that the tongue doesn't just shoot out and fly all over the place. It kind of comes out and comes back in. And in this case that they give here, the famous mantis shrimp appendage can strike at 31 meters per second and 25,000 G. So that, appendage, of course, is being sent out, but it's also being controlled by the organism, and then the anatomy is still intact as it's making that movement. It's worth saying that we do have a lot of faster movements, like uh, our whole body, if we were to sort of subject it to these high G forces, would be ripped apart. But we have things like if we throw a baseball or something, your arm moves, especially the fingers. They experience a lot more G forces in doing that locally. So it's not that we can't survive it. It's that it depends on there's a whole biomechanics of sort of a kinematic chain that happens that is sort of set up by the movement. So you're generating forces from the sort of the middle of the body outward, and then those forces are sort of being amplified as they go outward. And so it's the question is, what is the geometry of the structure that's being subjected to these g forces? Is it
Yes.
just kind of generic configuration, or is it something more um, specialized? So trap giants can strike their mandible at 64 meters per second and 100,000 Gs. So those are two examples from the multicellular world.
Well,
So
there's 5 million Gs. What's that? Over
this
the
one here.
Hydra.
Yeah. So this is okay. Actually, these examples from the multicellular world, though impressive, require specialized tissues and/or exoskeletons to achieve these ultra-fast movements. However, none surpasses the 5.4 million G acceleration of the nematocyst discharge in Hydra, which is ultimately a single-cell phenomena performed by specialized cells called nematocytes. So this is nematocyst discharge. Yeah, I think it's it's just a I don't know. Yeah, I don't know what that is, but I don't know the process behind it. Despite our fascination with the many macroscopic examples, the extremes in the cellular world remain largely unexplored. And then that's where they come in with their ultra-fast sort of phenomena characterization. So they actually kind of talk about Hydra here. So ultra-fast phenomena pose unique challenges for single cells, as they must store all required energy within their small size and deploy it precisely, like I was talking about, with different appendages moving towards a target. For Hydra nematocytes, the task is somewhat easier, as their discharge is a single shot, non-reversible event, so just shooting things out into the environment. And these specialized cells are supported by surrounding tissues. So these are, it's not like they're just being sent out and left to experience those G forces all on their own. There are surrounding tissues that support that. However, single cell organisms with ultra fast motility lack this luxury, as they must also perform all the necessary functions as a single unit. In addition, single cells must ensure their own survival and functionality after deploying an ultra fast mechanism, as extreme velocity and acceleration usually indicate the presence of extreme shear and bulk forces. So this is where you experience these different types of extreme forces during this process. So this is something again that you have to in some cases you have a tissue surrounding the cells that protect the cells, but in this case it's just the cell itself. And so you want to have a way to protect the interior of the cell and some of the mechanisms within the cell. So these constraints impose additional evolutionary pressures on organisms capable of ultra-fast motility. So in this review, we first present a quantitative map of single-cell organisms that demonstrate ultra-fast motility, including locomotion, contractility, volume expansion, and density changes. So these are all the properties. We then classify and quantitatively compare these behavioral phenotypes in terms of their acceleration, speed, area strain rate, volume expansion strain rate, and density change rate across the entire tree of life. We report these quantitative numbers either from the literature or from reanalysis of published data, and we conclude with a comprehensive review of the trigger mechanisms, as we had mentioned, and how those kind of correspond to evolutionary adaptations. This is the atlas. So you have this atlas, and it's just like different ultra fast cells, and they have like a legend here. So if we go in a little bit, maybe I can. Yeah, so they're numbered, and you can see that they're these different cells. They're kind of labeled by, sometimes it's genus and species, sometimes it's the genus SPA, which is like they can't define the species, but they know what the genus is. And so they're just these different, like, I see paramecium in here, and let's see, stentor, um, Altaria. I mean, a lot of things that you would see in the unicellular world. So that you have things with flagella, you have things that are kind of radially organized, you have things that look like a tree, you have things that are axially oriented, um, and other types of cells. Yeah, so these are all very interesting structures. Um, Definitely not what you might expect. Um, but yeah, so there are all sorts of evolutionary adaptations hidden in here. So just to give you an idea what we're talking about. And so the caption says organisms with ultra-fast cellular motility scaled relative to one another. Ultra-fast organisms, which come in a wide range of sizes, shapes, and behavioral phenotypes, are present in multiple major branches of the eukaryotic phylogenetic tree. This list includes organisms of fast translocation, contraction, extrusion of organelles, and even fast volume and area changes. So these are all things that, kind of ways in which they uh, execute these movements of translocation, contraction, extrusion, and then fast volume and area changes. So you can imagine like how, some of this stuff isn't like movements to and from somewhere. Some of it is changing its shape in response to the environment, but doing so in a very fast manner. So that's why, your uh, microtubules have to undergo this sort of uh, wide scale, widespread disassembly and things like that, because it's actually changing the structure of the cell. And it's doing it in an ultra fast way. So, yeah, they kind of go over some of these examples. Um, so there are one set of cells here. I think they're bacteria that can reach a swimming speed of up to 468 BL per second and 393 BL per second, respectively. They are thermophilic hydrogenotrophic methanogens, so they're bacteria preferring high temperatures near hydrothermal vents, and they live on oxygen or hydrogen and carbon dioxide, producing methane as a byproduct. Their swimming patterns are characterized by fast straight swimming and relocate to slower zigzag swimming when near a surface. So these are these kind of extremophiles that um, have to do things. They already have a lot of properties of kind of extreme adaptations to a hydrothermal environment, and hydrothermal vents at the bottom of the ocean where you're not only subjected to great pressures from the deep sea, but you also have to survive in these kind of really hot environments where synthesizing hydrogen and carbon dioxide and producing methane. So this is sort of also they're exhibiting these very fast movements. So this is kind of all part of the set of extreme adaptations. Another example of a fast bacterium is this species here, a predatory bacterium with a single flagellum capable of penetrating and replicating within bacterial prey. For strain HD100, this 1 micron bacterium can swim up to 160 microns per second. This, of course, is 160 times their size. Early hypotheses suggested that the species uses its fast motility to drill and penetrate the prey cell, but later genetic knockout studies showed that flagellar motility is not essential for prey entry, and thus plays a role only in efficient prey encounters. So that's an interesting mode of like survival, is being you're predating another bacterium, but you're much smaller than them. But you can actually move much faster than them as well. And a lot of that fast movement is to sort of encounter your prey. And this is in their flagellar, so they're moving their flagellum to make the movement. So it's a whole structure there of macromolecules that are involved in this. You were going to say something?
No, I just think it's interesting.
Yeah.
I don't know if I'd want either one of them inside me.
Yeah, probably not.
Just no.
These are hydrodynamic jumpers. So we classify organisms of swimming trajectories characterized by slower regulating swimming modes interspersed with sudden high-speed jumps as hydrodynamic jumpers. So these are like two modes of motion where you have swimming, which is slower, and then these high-speed jumps. So it's like whatever they might be encountering some prey or something, or they may need to move away from something uh, that is, maybe they're the prey. And that's these jumps within the water column. So many small ciliates have evolved the ability to perform sudden jumps, and they are collectively referred to as jumping ciliates. They include Halteria, uh, Mesodinium, Eskencia, Cycladium, Pleronema, Strobilidium, Eurotrichta, Euronychia, and Diaphoras, as well as weaker jumpers like Strabidium and Stylantia. So if we take Halteria granadella as an example, this 20-micron oligotrich normally swims forward at 0.2 to 0.3 millimeters per second, and that's 10 to 15 BL per second. I'm not sure what the BL measurement is, but that's what it corresponds to. But when disturbed by flow or a high concentration of potassium chloride, it performs a powerful backward jump at 3.2 millimeters per second, or 160 BL per second. The maximum normalized speed of jumping ciliates typically falls between 50 and 200 BL per second, with the Strobilidium species having the fastest recorded speed of 2.76 centimeters per second, or 361 BL per second, for a 76 micron cell. These fast jumps are enabled by exceptionally high increases in the cilia beating frequency from 105 to 260 hertz. So this is, of course, the cilia is driving this. It's beating really fast, and it's enabling these kind of fast movements. And of course, you need to have a specialized cilia for that. You can't just have these standard cilia. So that's a set of evolutionary adaptations there. Many dinoflagellates also exhibit hydrodynamic jumps. Examples include uh, Ancinella, Rafaria. Okay, so these are they're. Yeah, I'm not going to bother with it.
No.
There are a number of different species that do this, and A. grandifera also holds the record for absolute speed, swimming at 1,603 microns per second, while Y. unicinus leads a normalized speed at 1,572 microns per second. So, these are just kind of really fast speeds for swimming. Functionally, while these jumps have an obvious role in predator escape, organisms can also use them for ambush attacks or to escape their own nutrient-depleted boundary layers to enhance nutrient diffusion. Well, that's interesting. So there is this actual aspect of feeding where you just need to get out of your current area and go somewhere else, or you need to disperse the nutrients that exist in the water column. So that's interesting. Their fast swimming speed is also thought to help in reaching deep water nutrients. So this is just a way to sort of escape predation, but also to get nutrients. In addition to jumping ciliates and dinoflagellates, sudden jumps have been reported in Botosaltans, free-living kinetoplastid, in Resultamonus mostrupi, primitive green algae. However, no swimming or jumping speeds have been reported for these species. Although different species are capable of jumping, their jumps are mediated by the ejection of ejectosomes, and thus are characterized as cell protrusions or extrasomes. So I've heard of this before where cells will like explode or explode part of their body to move. Like there are these different explosive movements that you can observe where they eject a bunch of stuff into the environment and then they move. And that helps them move fast, like it's an explosion. So the explosion is very fast and sudden. It moves them at a very high speed. And this is, of course, what they call an ejectosome, which is, just saying that there's ejecta, and it's probably some set of biochemicals that gets ejected. Um, and so that's interesting that they bring that up. There are also elastic jumpers and surface-based motility, contraction without translocation. So this is so surface-based motility is where motility depends on a solid substrate, and this includes diatoms, and they have the surface-based motility. So diatoms generally lack cilia or flagella, can glide on surfaces by using an actin-myosin system linked to a secreted adhesive polysaccharide. So this is, we've seen this in meetings before where they secrete these sort of polysaccharides and other types of molecules that help them move in their environment on some sort of surface. Elastic jumpers can contract or recoil their long appendages instead of using ciliary or flagellar beating. So these are different types of ciliates that can do this. When their appendages contract, the cell bodies of Pseudotonadia and Tonatonia can achieve backward jumps of 653 and 1,470 BL per second, respectively. So that's kind of there. And then uh, electron microscopy studies show that its tail consists of numerous nearly parallel membranous tubes and septa connecting to small bundles of microfilaments, where the tubular layers are separated by hyaloplasm containing many mitochondria. So, of course, you need to have an energy source for this. Um, a similar mechanism exists in haptophytes when the microtubule array in the haptonema, which is an appendage presented only in haptophytes, switches its conformation. The haptonema coils back and causes a rapid jump of the cell body at 6 millimeters per second. Another ciliate, uh, uh, Zooxanthium pelagium, although it does not have a contractile tail, can contract the entire pseudocolony and achieve Medusa-like locomotion at 110 millimeters per second. These are,
That's a lot.
yeah,
Like that's very fast.
yeah, that's about 250 pseudocolonies per second. So, like, the size of the pseudocolony is, much smaller, and they can move over 250 pseudocolonies away in 1 second. So, yeah, it's fast. And this is all generated by, I assume, your typical, set of microtubule associated proteins and your typical
Is
mitochondria.
is that myosin by any chance? The,
Uh,
the calcium
well,
binding
calcium binding
microtubules
microtubules. Yeah, it doesn't.
protein,
I don't know. Yeah,
actomyosin?
maybe. It may be another form of
Is,
that.
yeah, well, how is it related to actomyosin?
Yeah, yeah. I don't know, but I mean, you can see this though. That muscles kind of do the similar things, right? Like,
Well, so do cell surfaces.
yeah, yeah. And the whole idea is that they're these structures that enable movements, sometimes very fast movements, but of course over short periods of time because you have to sustain that movement. And a lot of times it's a burst of energy that it needs to exert. So it's not like constant. Um, so yeah, there are a lot of like if you think about muscle fibers, they enable these kind of bursty movements. You have other mechanisms um, in actin and actomyosin as well where you can do that.
Yeah, it's associated with calcium.
Yeah,
Like
yeah, it's
it,
it's
it's Is that where it began? The calcium binding
It could be.
proteins and microtubules.
It
Like
could
that
be.
looks like very much like what happens in cells.
Yeah.
Um,
Yeah. Yeah, it's a calcium driven process, but yeah, it would be interesting to
and
see. I don't know if there are any papers on like thinking about that mechanism more in terms of like how it's generating these really kind of fast bursts of energy, but it'd be interesting to see.
yeah.
Because you see it in neurons too. Like you have action potentials that are based on calcium ions and things like
Mm-hmm.
that.
Yes, for sure.
Yeah. So then there's contraction without translocation. We now consider examples of organisms where contraction of either the entire cell body or its appendages is not used mainly for translocation. So this is like we were talking about translocation where they're moving in these bursts, but they're contracting either themselves or their appendages. Either these organisms are attached to a substrate with a stalk, or their motility is symmetric, and this results in little net translocation after a complete actuation relaxation cycle. So they have these cycles of actuation and relaxation. We saw some examples in figure one where they had like these
Yeah.
stalks and things connected together by some stalk. So this is kind of what they're getting at, and then things contract around that central stalk or that sort of thing. Um, yeah, so either these organisms are attached to a substrate of the stalk, or the motility is symmetric, and this results in little net translocation. So this is again where you have this symmetric motility as well. So you have two modes. So examples from ciliates include contractile heterotrichs like Spirostonum and Stentor, contractile sessilids like Vorticella, Carchaceum, and Zoothenium. Uh, for a review of the biomechanics of various contractile sessilid species, C33. So there's, if you're interested, you can go to citation 33 in this paper. Let's take these Spirostomum and Stentor as examples. Both are giant single cell ciliates that can grow to millimeter size and contract their cortical myonym to reduce their body length to less than 50% within 5 milliseconds. So this is like a reduction of body length just through this contraction process. For Spirostonum ambiguum in particular, this rate corresponds to a contraction speed of 0.2 meters per second and an acceleration of 15 g. So this is, kind of, again, these fast movements, but they're focused now not on spitting things out into the environment or moving in the environment, but contracting their own phenotype. Fast contraction has also been observed in various Heliozoan species. For species for species with outstretched axopodia, they can rapidly contract when in contact with food organisms. So these are these different projections that they have called axopodia, bringing the prey near the body's surface. Uh, the contraction speed of axopodia is estimated to be more than 2 millimeters per second. One special bentham Heliozoic species can contract not only its axopodia but also its stalk. The stalk, which is 300 to 400 microns long when fully stretched, can be shortened by 90% within 50 to 60 milliseconds, resulting in a speed of up to 10 centimeters per second and an acceleration of 10 g. Okay, so then you have these cell protrusions and extrasomes. So this is another trick in the armory of single cells, is the capacity to extrude parts of the cell or specialized organelles. Specialized exocytic organelles that can extrude materials at very high speeds are commonly referred to as extrasomes. So these are organelles that are shooting materials out. And so this is the extrasome. Examples include trichocysts from many ciliates, toxocysts from many predatory ciliates, dinoflagellate nematocysts, and then ejectosomes from cryptomonads, and then from all cnidarian species, and then discobollocysts from chrysophytes, and then so there are all these different mechanisms in different species and genera and orders that you can look at here in these citations. So they do talk about extreme volume and density changes here. And then they talk about ultrafast acceleration. And then they have this figure 2. So this figure 2 is, of course, where they have this table of different species and different genera. And you can see the different phenotypes. Some of them have flagella. Most of them have flagella. Some of them don't. But you can see that they're on this sort of scale, which is acceleration in meters per second squared. So you have these different, so the hydronematocyst, which we talked about, is really high in terms of acceleration. You have, organisms that exhibit lesser acceleration, and these are different mechanisms that we described earlier. So you can see this sort of continuum here. And then in B, you have this, not in terms of acceleration, but velocity. So you have some of the same genera here. You have hydronematocyst firing, which has a high velocity as well as a high acceleration. So velocity is measured in meters per second instead of meters per second squared. You have 3T3 cell migration down here at 10 to the negative 8th velocity. Neutrophil migration, then MMP ping pong motility. You have E. coli swimming. You have diatom jerking motility, which we've talked about in previous sessions. We've talked about that as sort of a high velocity mode in diatom motility. But in the scheme of things here, it's actually kind of in the middle. So diatoms aren't necessarily special in this regard, although they do have these ultra fast modes of movement. You have paramecium trichocyst firing, lacrimaria olor neck extension. You have jumping, you have contraction, so stentor contraction, you have jumping. Yeah, so you have all these different behavioral modes included in this graph. And then this is a nice graph. This shows, of course, you might be asking, well, if they exhibit these movements, that's fine. And, I would like to see it normalized to organism size, because there's probably a wide range of sizes represented here. So in C, we have cell size in microns versus maximum speed in microns per second. So this kind of normalizes different movement modes and organism types. This is ciliates, elastic jumpers in purple, ciliates, hydrodynamic jumpers in green, ciliates, swimmers, ciliates, walkers in blue, flagellates, elastic jumpers, flagellates, hydrodynamic jumpers, flagellates, swimmers, prokaryotes in pink, surface-based motility in blue, cell protrusion, extrasome in, I guess, light, wine-colored, and contraction without translocation in purple. So it's kind of, they use a lot of very similar colors, but the take-home here is that ciliate swimmers are kind of located in this middle section. You also have cell protrusion and extrasome in the same region. And some of the outliers include cell protrusion and extrasome out here. And you also have, let's see, you have ciliates walkers in the main area, and you have the surface-based motility down here. So what are we looking at? Well, we see that our outliers are generally in the cell size of 10 to the 1 microns. So you see this maybe a little bit less, a little bit more. And then the larger size cells, which are greater than maybe like 10 to the second microns, or maybe like, 100 microns and larger, they kind of follow this trend of maximum speed between 10 to the 5th microns per second and 10 to the first microns per second. So they're in that window. And so you can see there is a variety of things within that window as well, as you can see on the chart. So it's interesting. It's very, very, there's a lot to unpack. And certainly, you could make the case that there's a lot of diversity. So even within these categories, you have a lot of different modes of this happening. And even depending on if you normalize it for cell type, you see that there's a lot of variation in things. If we look at the figure caption, comparison of ultrafast acceleration and speed among various single-cell organisms. Most organisms can achieve maximum velocity of around 10 BL per second, justifying the definition of a normalized speed greater than 100 BL per second as ultrafast. So it's the 100 BL per second measure threshold. The cross indicates a discobolicist, which is an inferred number that has not been experimentally validated. And then we have our trigger mechanisms, our actuation mechanisms, and our dissipation mechanisms. So our trigger mechanisms can be chemical, mechanical, and chemically gated mechanical. So chemical being like these different chemicals, chemical effects like pH, digestive enzymes, redox potential, osmolarity, digestive products, and ion concentrations. And you might have these gradients in which the cell exists where they, react accordingly. You also have these mechanical triggers. So you have critical membrane tensions, critical strain rates, and then chemically gated mechanical, which is where you might have a prey-derived chemical signal signaling a sensory neuron, and then there's a synaptic release of acetylcholine, and then that's kind of talking about the triggers. Activation mechanisms is where you have spasmoneme, myosin, which is where you have these contractile heterotrachees, contractile cicelids, and eccantharians. You have cataclysmic microtubule disassembly, which you see here. You have decondensation of secretory proteins. You have purely hydrodynamic mechanisms such as jumping ciliates and ultrafast prokaryotes. Then you have osmotic pressure, which is shown here. Then your dissipation mechanisms, which is where you have single shot dissipation. You see this in the Cnidarian nematocyst where they release things into the environment. You have compartmentalization and separation from functional cytoplasm, which is shown here. You have external hydrodynamic dissipation. Examples include jumping ciliates and ultrafast prokaryotes. And then internal dissipation, where you have this sort of dissipation within the cell in this species here. Okay, so I think that's enough for that paper. It's a lot of nice figures. It's a very hard paper to get through if you don't know much about the biology, I guess, because you have to go look things up constantly. But no, I mean, I think the take-home lesson here is that you have these different mechanisms for these ultra-fast movements. They occur under a wide range of conditions in a wide range of species across the tree of life, and perhaps the mechanism specializations are very different in different, even producing the same type of movement in different species and genera and orders that you find them in. So, any comments or questions on that?
Okay. Yeah, I said they should try using tensegrity structures
Oh yeah,
and studying that. It'd be a nice master's degree.
yeah. You mean the things that kind of make these things possible or hold the tissues together as they make these movements? I guess in this case they're just single cells, but they could still have tensegrity structures of like.
Yeah. Well, like I said, from macroscopic, including our own bodies, to microscopic, including internal cellular mechanisms, are tensegrity structures, where they can be made into tensegrity structures because they're in tension. They're held together in tension quite often. So that's why it's a tensegrity structure.
Yeah.
Especially if you have microtubules involved.
Yeah.
It's a good project.
Yeah. Well, yeah, Prakash always has interesting things to say about different aspects of biophysics, so that's good. I don't know, Morgan had anything you wanted to say, or there he is. Oh, he's home today.
Yeah, I just got dropping my. Hello.
Yeah.
Sorry. So I'm not sure the context, but I was trying to listen in when I could.
Yeah. Okay. Well, we were just talking about paper about different cells and how they make these ultra fast movements. And they had this cell atlas that they built of all these different cell types that can make these really fast movements, like either like through explosive movements or through sort of collective movements using flagellum and other types of mechanisms. And so it was really interesting because they kind of cover a lot of like different species in different parts of the tree of life and seeing kind of how those are generated. And there are kind of connections, I don't know if you missed it, between like the sort of calcium signaling you see in neurons that lead to action potentials and then some of the calcium mediated signaling that leads to actin producing some of these movements.
Well, yeah. So I'm sure you've seen this in Michael Levin videos where he talks about calcium as it's just cellular computation.
Oh, okay, yeah.
Yeah, like he's trying to find a word which just makes it just generic.
The trijunctions in cells also use that mechanism. Actin, myosin.
Oh, all right.
Yeah. And calcium. You have to have calcium and actin and myosin and ATP. And then you got movement.
Yeah.
Which, you couldn't imagine that being a big day in cellular evolution.
Yeah.
Well, it's interesting that these primitive or not primitive, I don't know, smaller organisms use that as well.
Mm-hmm.
That's why I was asking about actin and myosin associated with the calcium and the proteins that they're talking about. Like, what proteins are those? You said proteins. Well, actin and myosin could be considered proteins, I guess. Well, so, or is it different? That's a question.
I've been trying to get through all the papers, all the Levin papers that are listed on the Institute for Computationally Designed Organisms.
Okay.
And I think it's the most recent. Actually, I don't even know if this is the one that's. I think this is the one that's missing. Maybe it's still like bioarchive. But certainly like the 2025 paper, super interesting in terms of the genetic expression, right? So the transcriptome of the anthropods is super strange and interesting, right? So it's like, remember, it's epithelial cells, right? That have self-assembled into this glob, but this glob is using cilia like a single-celled organism to move, yeah, and have these behaviors that are super striking. And again, this is 100% human cells, no gene edits, etc. And it's the paper describes the transcriptome as interesting in that, and I don't know how they derive this, but they want to say very ancient profile.
Okay.
And I wonder. I mean, I think it would be really fascinating to get at both, how they would come to that kind of conclusion, and as well as, kind of speaking to Susan's, what was it, Actin, Myosin, ATP, and calcium.
Calcium. Yeah.
Like how much of that particular cluster is a part of that ancient profile. Anyway, a really fascinating group of results in this last paper.
Can you send those
Absolutely.
references?
Yeah, sure. Can I put them on the DevoWorm Slack or?
Yeah, I can send it to Susan then from there.
Okay. Okay. I've got your email. I'm sure, Susan. If you want, if you prefer that.
Okay. Well, I sometimes use Slack terribly.
Okay.
I'm not very good at it.
The other really interesting, and I think I only caught this from he's been doing this symposium on platonic spaces. It's like anyway, look it up. It's not as philosophical. I mean, it's still tied to his experimental work, but I think he was trying to make the case that these cells can hear, or the anthropods can hear.
Oh,
and
hear?
And by that, yeah, and by that he means I should every time he uses language I need to be careful.
Yeah.
Because that it can feel pressure
Right,
waves.
Yeah.
Okay.
And I think the way he wants to use them, it can like feel pressure waves and act on them,
Okay.
or
Yeah.
respond to them. I mean, I think part of that response is needed so that he can determine that they are hearing. Right. So I think because remember with the planaria, he when he caused the eyes to grow in a new spot, right? He had to create some apparatus to test whether they were functional.
Right.
And he was doing the same thing with the anthropods and sound.
Well, anything to survive. I mean, if you can feel pressure waves, that would help you survive.
Yeah.
So.
Yeah. So and like again, like what what's interesting, right, is that like this is the same, like you can take epithelial cells and you can get their transcriptome, but in this context, they've completely completely changed their expression profile and have made themselves new functionality. I mean, again, like not new functionality, but like they've like turned on functionality.
If you take them out of their environment, you will change their genetics.
Absolutely.
Their operating genetics.
Absolutely. Yeah. And but it's I think he's, again, like, I don't know how, I mean, I think there's something obviously really interesting in terms of finding this set that it wasn't, finding this kind of latent expression profile.
Yeah, that's interesting. I'd like to know what it was exactly.
Right. Anyway, so yeah, okay, we'll follow up. It's hard to make time for this because it's like I'm still not quite sure under what kind of project title do you put this, right? Like,
yeah,
it's anyway. It's like, do you put this under a kind of like origin of life?
right.
I mean, it's like,
yeah, actually, you could.
Right.
It's a step up from the very beginning, but
Right.
it's in there somewhere.
I just want to add, we're going to be covering the new book by Blaise. It's like Blaise
Arcus Aguera or something. Yeah.
Yes. Thank you. What is intelligence?
Yeah.
And
have you got the mechanical intelligence in there?
It's
where they built
got
circuits?
A lot. It's got a lot, and I bring it up not so much about the book, but just driving kids to activities and things like that. Like, I've been listening to as many YouTube videos as I can find, and he's got a lot. I mean, going back a long ways, and over a wide variety of topics too. So
any tensegrities? I have trouble finding anything on tensegrity on YouTube that explains more than just the basics. There isn't anything really.
Yeah. I mean, no, tensegrity. That is it's a hard one to find. Good educational material on. I would say I don't think he covers that, but I will say that like his work is super eclectic, and like he'll deep dive on something about creativity, music, certainly covering things about DNA and I mean gene expression and things like that, as well as I mean it's somewhat Levinish in his broad set of interests. And anyway, it'd be good though. Thank you, Bradley, for the. I forget who it was. You sent a link about somebody having some questions about the book, and
Oh yeah. There was someone who responded to like some of his stuff at A Life conference.
Okay. Yeah.
Yeah.
He had a really interesting talk at the it's called the Sainsbury Welcome Center.
Yeah.
It's like the neuroscience. Yeah. Anyway, that was really interesting. That was a particularly interesting one, and anyway, just coming back to this kind of fundamental mechanisms of agency, right?
Right.
Which are and that of course goes to single-celled organisms as well.
Yeah.
Anyway, yeah. Let's follow up on that last paper of theirs, of his. Of Levin's on the anthrobots.
Oh, okay. Yeah, we'll do that.
Yeah. If Blaze has, if he has some short papers, that'd be great.
Yeah.
But yeah, we're gonna cover that book in November 18th. So
Okay. What is this?
This is San Francisco Cognitive Science Reading Group.
Okay. Yeah. Right. Tell me about that.
Yeah. We've done a couple Levin papers or Levin co-author papers, but the guy who runs the group actually was on Blaze's team for 5 years.
Oh, okay.
So it'd be interesting to see if he's got any insight. He's a Google DeepMind supposedly working on like ML music and art, which I think is I still don't know what that makes you a product manager of.
Yeah.
Yeah, I should try to get employed with something to do with my art and science. The pictures in the background here are my.
Okay.
Yeah.
Yeah.
Lovely. Yeah. Well, these days you gotta have work AI into the narrative.
Yeah.
I mean, yeah. If you want funding.
Yeah.
Yeah, funding.
If you've got a startup.
There's always some buzzword though, like.
That's right.
Transcriptome.
You gotta fit in as many buzzwords as possible.
Whatever. Yeah.
Okay.
Yeah. All right. So that's great. Yeah. So let's follow up on that stuff and.
For sure.
Yeah, we'll see. Maybe in subsequent meetings we can unpack maybe some of that.
Okay.
Right.
Get registered for the Active Inference Symposium.
Oh yeah. All right.
Sounds
All right.
good.
Well, it was a good meeting. Thanks. Have a good week.
Take care.
Take care.
Bye.
Bye. As promised, this is the other paper I had for today. This is a paper on self-organized biotectonics of termite nests. So this is, of course, about self-organized insect nests and self-organized social insects, and then biotectonics being a mechanism for changes in the structure of that nest. So a lot of interesting people on this paper. Guy Thurlows, who has done a lot of stuff on swarm intelligence and social insects. El Mahadevan, who has done a number of things in biophysics, recently has done a lot of work on Lagrangian coherent structures in biology. And then Alexander Heyde, people from Harvard, people from University of Toulon in France, and then you have people from Harvard, both in organismal and evolutionary biology and physics. So the abstract reads, the termite nest is one of the architectural wonders of the living world, built by the collective action of workers in a colony. Each nest has several characteristic structural motifs that allow for efficient ventilation, cooling, and traversal. We use tomography to quantify the nest architecture of the African termite, Apicotermes laminae, consisting of regularly spaced floors connected by scattered linear and helicoidal ramps. To understand how these elaborate structures are built and arranged, we formulate a minimal model for the spatiotemporal evolution of these hydrodynamic fields, mud, termites, and pheromones, linking environmental physics to collective building behavior using simple local rules based on experimental observations. We find that floors and ramps emerge as solutions of the governing equations with statistics consistent with observations of A. laminae nests. Our studies demonstrates how a local self-reinforcing biotectonic scheme is capable of generating an architecture that is simultaneously adaptable and functional and likely to be relevant for a range of other animal-built structures. So it looks like they're hitting on a number of themes here. What's interesting is it looks like they're going in the direction of Lagrangian coherent structures. They talk about this sort of minimal model for spatiotemporal evolution of three hydrodynamic fields, and they're using mud, termites, and pheromones as these fields that intersect in these structures. We're linking then environmental physics to collective building behavior, so you have the collective activity of termites, but also the resources that are involved in that. So if we get into the paper a little bit, they talk a little bit about the termite genus Apicotermes that they're talking about here. They typify this architectural complexity that you see in a lot of social insects. You see bees' nests and termite mounds. These termite mounds are native to the savannas and forests of Africa. They construct small ovoid nests of just 20 to 40 centimeters in diameter, located 5 to 50 centimeters underground. The nest structure must be efficiently constructed to be capable of passive ventilation and cooling, while remaining habitable and transversible by the termites within. And so they talk about sort of how there's a lack of centralized control, that there's no way that any one termite could have a sufficiently broad cognitive map that could provide this sort of a master plan of this architecture, an organizational structure that allows everyone, that sort of directs all the other termites to coordinate this colony. So this has to emerge from collective behavior from individual termites doing local tasks or contributing to small parts of the architecture and then the architecture emerging from that. So this is kind of the idea here, is that termites acting only on their local conditions produce global order. So they get into the idea of stigmergy. And so stigmergy is where you have this sort of collective behavior, but it's expressed somewhere in the environment. So it's laid down in the environment, and then that environment is used as a feedback signal to the rest of the collective to continue building upon that. So if you think about, like, if you think about ant colonies where they build ant trails, and the ant trails are determined by pheromones. So the ants will explore their environment and lay down pheromones. And as they lay down pheromones, those concentrations will tend to aggregate in paths that go from some source to, like, maybe a nest to food. And so they, over time, as they explore, they tend to take similar routes across the environment, laying down their pheromone. And as that pheromone trail grows in concentration, then that pheromone trail gets reinforced by ants kind of being attracted to it. So you can see the stigmergic mechanism, this feedback between things that are laid down in the environment, and then cognition and recognition of that signal, and then reinforcement of that signal through more pheromone being laid down. So they talk about the principle of stigmergy, defined as the spontaneous but indirect coordination of agents, stimulated to action by the trace of previous actions left in the environment. And this is implicated in what they call the morphogenesis of large structures that enclose the nest. Over the long length and timescales of overall mound structure, the small internal tunnels and chambers within the mound influence the mound's material porosity, mediate ventilation, and explain the coarse features of the mound, such as its shape, size, and temporal evolution. So they actually were able to collect, scan, and then analyze the structure of these nests. So here's some of the images here. These are the scans. They would take, like, the nests out of the ground. They would do like a 3D scanning of it. They would scan the inside and the outside. capture this sort of form. And then, once it was digitized, they could then analyze it, manipulate it, whatever. So they've identified these different nests. The one is called MIMO 80, and then the other is MIMO 14. So they basically have two nests that they identified that they scanned in, and they looked at the spacing and arrangement of the floors and ramps. You can see the floors are here, these different levels, and then the ramps kind of connect the floors so that the termites can move up and down the nest. So for each nest, we assembled a series of 359 and 177 vertical slices respectively, measuring the thickness of the floors, the vertical spacing between floors, and then looking at sort of the other properties of the nest. They've noticed that there was a consistent arrangement of floors. The positioning of ramps was measured using the horizontal distance from each ramp to the nearest ramp on the same floor. They found that this was actually ordered in the sense that it deviated from a null model of randomly scattered ramps. So you could ask, are these ramps just kind of randomly scattered across the nest, or are they at regular intervals? And it turns out that they're more at regular intervals than random, which suggests that there's this sort of architectural style that they pursue. Even though they're not doing this from a master blueprint, they're doing this from sort of their collective behavior. They're able to do this quite well. And so now they have this minimal model of biotectonics. So we turn to a series of observations to motivate a minimal continuum model from nest construction. So we model the spatiotemporal dynamics of three fields that depend on location, denoted by the vector x and time t. So the nest material density, which is uxt, scaled from no nest material when u equals 0 to fully compacted when u equals 1. So it goes from 0 to 1. And the nest material density is then uxt, where u can range from 0 to 1. The termite worker density is nxt, and the pheromone concentration is pxt, both in units of mass per volume. These three fields jointly evolve according to a sense of conservation laws expressed as the following differential equations, and they show that in the appendix for the nest material, termite workers, and pheromone levels. So they're working from conservation, from first principles, and they're watching these three fields evolve according to this, across this where this architecture would be. So this is figure 2. This is the statistical analysis of nests. So this is where we see the different nests, MIMO 80 and MIMO 14. You can see the differences between them, the ramps and the levels. And then what they're doing here is they're analyzing floor thickness and floor spacing, and they're looking at distance in millimeters versus frequency in percent. So the distance and their frequency. So how often do you see structures of this size? And so you can see for floor thickness and for floor spacing, you get these sort of unimodal curves. I wouldn't call them normal distributions, but they're pretty close to something that has a mean and a variance that suggests that there's some sort of mean behavior going on in the building process. And you see this for both nests. And then maybe for this nest a little bit more than for this one, but still. Then that's B. A is, or the nest. B is this graph, distance versus frequency, and then C is where you have distance versus frequency for the same floor or adjacent floors. And in this case, they're using a function, a continuous function, showing this, where you see this for the same floor, you see this for adjacent floors, and you get this sort of, you get this difference. So for adjacent floors, you have this difference between the same floor. More in this nest here in MIMO 14 than MIMO 80, but you still see some sort of difference there. So actually, they give the differential equations here as well. For nest material, you have this equation here for one. Termite workers have another equation, and then pheromone is a third equation. So for these differential equations, these two symbols T and Z are the differential operators in time and height, respectively. The gradient symbol or the nabla symbol is the three-dimensional divergence operator, and then the nabla dot is the three-dimensional divergence operator, and nabla itself is the three-dimensional gradient operator. So you have a gradient and a divergence operator in the equation here. So like in termite workers and in pheromone, you have a three-dimensional divergence operator. In the pheromone and termite workers, you also have the gradient operator in different locations in the term. So what that means is that nest materials don't have a gradient, but termite workers and pheromones have a gradient. Okay, so in equation 1, this reflects the addition and removal of the nest material, so it's a sort of just this differential rate of the substrate. The poroelastic diffusivity of dirt G captures its capacity to settle under the influence of gravity. This term breaks the rotational symmetry of the model equations and establishes a defined vertical orientation for the nest. Equation 2, which is termite workers, reflects the dynamics of termites carrying dirt or not carrying dirt with a flux that has two components. First is an effective diffusivity that is proportional to the amount of open space, and a chemotactic term that drives termite workers into open and low density nest regions with chemotactic coefficient chi. So remember, they're being guided by chemical gradients. So they're using pheromones to guide themselves around the environment, to leave signals in the environment that they then follow and push dirt. So basically, it's a communication mechanism. The final term represents switching between N and eta plus and eta minus, with the pellet size K denoting the average amount of dirt transported by a worker. So again, we have this pellet size. We have switching between different modes of that. And then finally, equation 3, which is the pheromone, reflects the dynamics of pheromones as they diffuse with the diffusion coefficient and are secreted into deposited soil level H per pellet and degrade through evaporation at a rate. We note that the pheromone is relatively non-volatile and thus not advected by the ambient fluid, since the subterranean nests of apicotermes do not typically have noticeable temperature gradients. And so then we have, for the rest of the model, we have these two equations here, building rate and removal rate. So again, we're building and then we're removing. And so, yeah, we have this figure 3, which is the biotectonic model that predicts floor spacing and ramp emergence in termite nests. So basically we have the nest material, which is this UXT. We have collective motion of the termite workers, NXT. We have signaling via the secreted pheromone, PXT. And then we have this templating process where it gets deposited in terms of the stigmergy as the nest. So the nest material is taken by the termites, and the pheromone is used as the communication signal, and it's deposited in the nest. So the pheromone tells the other termites where to lay the pellet. They lay the pellet, and then that signals more termites to lay other pellets in other locations, and then that just kind of works its way without any sort of centralized guidance or architectural guidance. The architectural guidance, I guess, is limited to the pheromone deposit and the location of that pheromone deposit and the existence of a nest and how that builds up to larger and larger structures. And so we can see this like one snapshot from the nest where you have these levels and ramps and how gravity influences this and how removal influences this and how deposition influences this. There's this building rate versus pheromone concentration. So the building rate, as that increases, pheromone concentration increases linearly. However, the removal rate is flat and has no effect on pheromone concentration has no effect on removal rate. Then this shows kind of a three-dimensional model here of edge dislocation and screw dislocation, and then how there's this slip plane. So you get these ramps by there being a dislocation of the edges. So they have this model where there's sort of a slight dislocation in the depositing of the pheromone in this graph representation, and that graph or that displacement of the pheromone translates into a three-dimensional plane in this sort of slippage, which is then the ramp. And then you can see this in this image of the mound, the termite mound, where you get these ramps that result from this slippage. It's a slip, what they call a slip plane. And so they were able to simulate several of these nests, and that's figure 4. And then that's the paper. So it's a very good paper. There are a lot of sort of implications of this. A very good model, I think. So this model allows them to capture 2 geometrical and topological features of termite nests, namely the regular vertical spacing of floors and the horizontal spacing of ramps on the same or adjacent floors, as well as the month-long timing of nest construction process. So this is just where we have this feedback loop of pheromones and building, and how that can be explained by stigmergy and this sort of emergent process.